All right. Hello, everybody. It's my great pleasure to see you all back at the VMOL seminar. And yeah, today is especially awesome because we have Meren giving the seminar today on Anvi'o, which I think is perhaps one of like the computational platforms to consider if you do microbiome data science. And yeah, just to give you a very brief background. So Meren is a computer scientist by training. He went to a small university in Turkey to do his undergrads at Kanakkale Oneski Mart University before he then decided to come over to the US to do his PhD at the University of New Orleans. And I think that's where he got kind of like excited about the microbiome and like the underlying computational needs we have. He then moved on for a postdoc at MBL in Woods Hole, and then became a assistant professor at the University of Chicago before then the, I think, University of Oldenburg. And Alfred Wegener Institute got very lucky to hire him as a full professor there, where he is now, I think, since like 2 or 3 years. So yeah, without further ado, it's our great pleasure. Thanks so much for doing this, Meren, and I'm looking forward to your seminar.
Well, thank you very much for the invitation, and I'm very thankful for everyone's time here in advance for coming to a seminar that is going to be given by a computer scientist who studies microbial sequencing data. So I guess it couldn't be any farther from your expertise and your interests in general. But I'm hoping that I'm going to turn this into a relatively useful experience for you also by introducing the software platform that perhaps could help you in your research going forward as you move from perhaps amplicon sequencing to more genomics, pangenomics, and so on and so forth. So today I'm going to be talking about Anvi'o, and I use this analogy of a kitchen in a world of restaurants, and I hope that will be slightly more clear as I go forward into this talk. Just to say a little bit about our own research program here that is one of the byproducts of which is Anvi'o, is that our research program brings together expertise in computer science and microbial ecology, and we use integrated omics and wet lab strategies to generally study microbial diversity and metabolism, microbial population genetics and evolution, and microbial physiology and translation dynamics. I am not going to be talking about these things, but I would like to say, in a very general sense, we are trying to understand strategies by which microbes respond and adapt to environmental change, which is probably something that every microbiologist could say about their research. And just like everyone else, we're also relying on integrated omics strategies to achieve these perspectives we're interested in. And these are extremely common strategies now in the modern era of microbiology, as you all know, metagenomics, single-cell genomics, pangenomics, phylogenomics, different data generation technologies, different data analysis technologies, and there is truly an interconnected nature to these things, of course, because unlike you who study metabolites, we are extremely lucky because we have a convening a piece of data that we call a genome. So we can bring everything back together from transcripts to proteins to DNA sequences from the environment and so on. So we have this very luxurious environment to make sense of stuff. But even then, actually, when you look into the ways by which people study omics data, there are significant challenges. And these challenges were much more prominent around the time we started Anvi'o as a project. And some of those challenges, in my opinion, included the lack of interactive environments for life scientists to perform in-depth analysis of omics data, where you couldn't see really what you're studying. These extremely complex data sets were very difficult to visualize and allow people to get their hands dirty with them. There were no easy ways to combine datasets of different nature. For instance, putting metagenomes together with metatranscriptomics or pangenomics in a single intuitive display was not quite straightforward. And ability to share or communicate findings that emerge from complex data has been a challenge, and it still is a challenge, I would say. We end up generating these figures that come from, in some cases, billions and billions of sequences, and we summarize them into a single Venn diagram or, you know, a hierarchical clustering result or PCOA plot. And of course, the underlying data could lead to many more insights than what we're presenting in a static figure that appears in a paper. But there are no ways to really communicate what else could be learned from the data as a function of the complexity we're dealing with the data. And I always bring this up. This almost is never relevant to people who are in the audience, but I find it to be extremely important. There are no ways to access the reasonable data structures for mathematical or algorithmic mining of complex omics data without having to deal with boring steps of common workflows, because we don't have a common environment to study, for instance, genomes where we know where genes are, where we know what functions are encoded by these genomes. Every single person who wants to apply to a new idea to genomics need to start from gene calling and making sense of BAM files and so on and so forth. So this is, to me, it's very similar to the earlier days of computation where there were no operating systems. So if you wanted to use computational resources to solve a problem, you needed to start from the operating system. You had to create the universe to make a pie. And then now we have operating systems on which there are libraries that programmers can use. So we don't have to reinvent the wheel. You don't have to think about how am I going to communicate with the keyboard input. It is provided to you. So that's what I mean when I say we are lacking these environments for supporting advanced research. And another thing that I always found extremely challenging is very difficult to put in words. So I try to actually use ChatGPT to come up with a conceptual image. I asked ChatGPT to demonstrate a concept where complex data go into a black box software, which turns all that complexity into a simple summary, such as an Excel spreadsheet or pie chart. So all the complex data go into something that you know very little about, and it does its magic, and then you get an Excel spreadsheet. And I didn't like any of the visualizations, so I will not show you anything apart from this prompt, but I hope you all have an idea now in your mind and a visual representation of this concept. So this is very common because this is easy to do. This is very little investment from a technical point of view to create software tools that take the same kind of metagenomic data and give you a list of taxa in it, or take a bunch of genomes and give you the gene clusters in it, and so on and so forth. So this is easier to implement, and that's what we have been doing in the field. It's not a blame, it is just a reality. But this is similar to going to a supermarket and finding only frozen pizza, that you get the same exact thing and then you end up eating the same kind of food. And if you are someone who wants to cook their own meal, you have a very specific idea about what needs to happen. You now need not frozen pizza, but the individual ingredients. You need a kitchen environment where you can actually bring those ingredients together and then implement complex ideas that perhaps were not thought by the farmers who create those individual items in your shopping cart. So Anvi'o was kind of an attempt to establish those kinds of principles, at least starting with our research and hopefully for the community. So Anvi'o is an open source community-driven software platform for integrated omics. And even though it was an idea and had slow beginnings around 2015, as of today, Anvi'o has over 175,000 lines of code, all of which are licensed with general public license, very communist. GPL license ensures that the code belongs to you as much as it does to any of the Anvi'o developers. So there is no ownership, there's no copyright. You can take our entire code base, rename it, and then say, this is mine, and there is nothing wrong with that. You can build a company around it and sell services using our code. There is nothing wrong against that. So you own the code as much as we do. So it's another seminar perhaps to discuss how much pressure we got back from other software initiatives in our field, because they no longer could use our code in their own platforms, because there is only one aspect of GPL that makes it less desirable than MIT license or FreeBSD license, and that is when you use GPL code in your code, you have to make your code available to everyone for the entirety of your platform. So if you have a platform that uses, for instance, a closed source software, and then you want to use pieces of Anvi'o, then you need to open source that closed source software for everyone to be able to see its code. And that is, of course, not applicable to every, I don't know why, but it's not working the same way for everyone. So it is GPL license, it is yours, whether you care about it or not. There are about 150,000 words worth of tutorials online. So just to make sure that people don't need us to do their own science. So they can do everything without coming to us and hopefully find their own way around the platform. It is quite a robust and extensible design. Well, the design is 10 years old now, but still it's solving our problems. We can add new features to it every other year and still supports addition of new features without having to replicate code in the code base. It is integrated visualization. I will show a few examples of that, that helps people to see what's going on with their data and play with it. And it is a community-driven effort. And I'll get back to this towards the end of my talk, just so I establish this point a little further. So Anvi'o comes with a bunch of small programs. Anvi'o, in fact, is nothing more than a library, a software library for which we write clients. And then we implement new ideas that use that library in different ways. That's why the kitchen analogy actually works. So this is a screenshot from recent times that shows Anvi'o programs that are accessible to you in your terminal. And there are about 160 of them, I think. And each of these individual tools are doing individual things. They are not giving you entire workflows, but they do one thing. One of them turns a FASTA file into a contigs DB. The other one takes a BAM file and creates a profile DB and so on and so forth. So you don't need to look at these names. They don't matter, but essentially there are many small tools that do one thing and one thing only. And then you can now connect them to one another to actually create workflows that we did not think about, for instance, while creating these things. In a way, this is similar to pieces of Lego. With the interlocking system in place, you can create different combinations. And I can present Anvi'o to you as a way to create a spacecraft, and then you can make a unicorn out of it, kind of thing. So, of course, looking at the list of program names like this don't help much about the interconnected nature of these things. So one thing Anvi'o can do, and it took a lot of coding actually, is this introspection. It knows concepts it can work with, like BAM files, FASTA files, gene clusters, phylogenetic trees, and so on. And it knows programs that can consume or provide those kinds of concepts. And then it turns into a network like this. So you can see all the connections. Of course, I'm not going to explain any aspect of this network to you. This is just a visualization of all concepts and programs in Anvi'o universe. But I will just say that every corner of the ecosystem has tools and small things that could be applicable to anyone's research who comes close to pangenomics, phylogenomics, metagenomic read recruitment, population genetics, and so on and so forth. For instance, if you have a BAM file, there are many ways to get into this network. And it's not important whether you fully understand this network or not, but I just want to use this as an example to show how Anvi'o workflows go about. One of the ways by which you can enter into this network is a BAM file. And BAM file is the standard file format that help us describe read recruitment mapping results to a reference context. And if you have a BAM file, there is one program that operates on BAM files in the Anvi'o network, and it's called Anvi init BAM. And when you run Anvi init BAM in a BAM file, you get a proper BAM file, which now is recognized by Anvi'o environment as a proper sorted and indexed BAM file that other programs can work with. And once you have this, you have multiple options that can run things on this. One of those options is to profile it. You can profile it to understand read recruitment coverage statistics throughout the entire sequence context, depth of coverage, breadth of coverage, and so on and so forth, single nucleotide variants, single amino acid variants, what kind of population heterogeneity we're able to capture through these metagenomic reads in the environment in the context of our reference sequences and so on. And when you run Anvi profile on that, you get a bunch of things, one of which is the raw variability data for population genetics analysis, where you learn about the nucleotide variation in the environment for your reference sequence. And once you have that, you can do a bunch of things, including running this other program in the network, Anvi GAN variability profile, that allows you to filter certain nucleotide positions, focus on certain genes if you want, or metabolisms or genomes and so on. And now from that, you get another concept data, which is process variability data. And if you have that, you can do a bunch of things with that, one of which is Anvi display structure, from which you can get an interactive interface. And you would literally be looking at a figure like this, where a gene in your genome how genetic variants found in a marine sample looks like in the context of the predicted protein structure for your gene. So this entire workflow is basically traversing this network with known or new workflows or ideas. And it has quite a steep learning curve, I have to admit. But once people get a hold of it, actually, it becomes extremely powerful in their hands. But essentially, rather than serving as a monolithic program that enables one specific analysis path that maps every input dataset to a specific output, Anvi'o aims to give the driver's seat to its users with this modular and somewhat frustrating structure. And of course, as you can imagine, implementing and maintaining a platform like this that is getting closer to 200,000 lines of code is not easy. And as I mentioned at the beginning, we're not actually in this for the joy of developing tools. We are scientists ourselves. And this tool and others coming from our group is just a byproduct of our needs that we try to share with the community. And one of the ways by which we can continue doing this is because Anvi'o, progress in Anvi'o takes place through people's science with it. And for instance, this is a recent, relatively recent paper. The original paper was from 10 years ago, so we published this as an update to that. And all these authors here have contributed to Anvi'o one way or another. And most of these authors at the beginning are PhD students in our group or in other groups that used Anvi'o to go after certain ideas. And then they wrote their paper, but the code that we worked together to integrate in Anvi'o became available to everyone immediately. I'll give you a few examples of the kind of research that we were able to accomplish with Anvi'o very quickly without going into details. For instance, with Evan, we used Anvi'o to integrate metagenomic and in silico protein biochemistry to map environmental sequence variants on protein structures to be able to elucidate, at least attempt to understand the targets of distinct evolutionary processes as a function of changing selective pressures that affect marine systems. So we brought together predicted protein structures with environmental variability to tease apart adaptive processes of evolution from neutral processes of it. And with Alon Shaver, we used Anvi'o to integrate genome-resolved metagenomics, pangenomics, and phylogenomics to show that human dental plaque likely served as an intermediate medium for some soil bacteria to adapt to host-associated habitats. It's an entirely different kind of research where we were able to show that your plaque that you want to get rid of every night for a good reason may have served as an important springboard for some environmental clays to find niches in host systems. And with Emily, we used Anvi'o to study the ecology of a single cryptic plasmid with 2 genes only and show that it is likely the most numerous genetic extrachromosomal entity in the human gut with seemingly absolutely no reason whatsoever. So we spent 5 years studying this one plasmid that all of you have, you on this call, all of you have it. And when you have it, like 95% of people have it in the Western world. If you have it, it's the most numerous extrachromosomal genetic entity in your gut environment. So weird. But Anvi'o enabled us to do quite extensive analyses, put this plasmid in the context of all global metagenomes and so on, and we managed to write a paper on that. With Eva, from whom you will actually hear in a few weeks, around a topic that will be more of an interest to you, hopefully. We used Anvi'o to study microbial metabolism in the context of human gut microbial ecology by developing tools that could help us understand the metabolic makeup of individual genomes and metagenomes together. To talk about the determinants of change in biodiversity, metabolic determinants of change in biodiversity in environments under stress. And with Blair, this is my last example, we did something very interesting. We focused on the diversity generating retroelements, which is a genetic mechanism that is not very well understood, relatively recently discovered, that generate completely random sequence patterns in specific regions in microbial genomes and yield haplotypes that partition population as a function of environmental pressures. For instance, here we showed a trichodesmian population and how very highly variable regions in its genome due to diversity generation retroelement activity partitions these populations as a function of biogeography. And to accomplish this, this is a very unusual research. I haven't seen anyone do anything like that. We designed in silico primers for DGR targets, recovered short reads from FASTView files, aligned them, performed oligotyping analysis on these sequences to resolve ecological patterns, and then we were able to investigate the amino acid divergence patterns across environments. The only reason I'm mentioning this, I know that this research does not make any sense to you. All I want you to appreciate here is that this is very difficult to do, and it's very random also. No one does these kinds of things. But what I want to highlight right after that is the fact that Anvi'o is not designed for this research. We haven't designed Anvi'o for this. And yet we could do this without adding a single new utensil into the existing Anvi'o kitchen. So by just using the tools in the Anvi'o portfolio, we were able to do something this random that, you know, and I find it's kind of interesting and exciting. And this also is why it is difficult to present Anvi'o to new people, because it is a useless space when it's empty, but can be quite powerful in the hands of someone who has unique needs and require full control over their questions and data. If giving your metagenome to a taxonomic profiler and getting a spreadsheet out of it with taxon names, if that is enough for your research, you will never need Anvi'o anyway, so you don't need to worry about it. But if you need to be much more specific about the phylogenetic breadth of the things that you're interested in, or if you want to add new sequences into the mix, or if you want to understand or control which genes are used for taxonomic profiling of your data, and if you want to see which taxa are more heterogeneous in the environment than other taxa, instead of just seeing a single name in the exact spreadsheet, then this kind of a platform becomes a little more useful. And so we apply, and I hope these examples clarify to all of you that the only reason a group like mine that is led by a computer scientist who has no training in biology, we were able to pull this off. We published papers on plasmid biology, we published papers on evolution, we published papers on metabolism. The only reason we could pull this off is because we have this powerful tool in our hands that allow us to learn from data. And this also highlights an important aspect of using platforms that are not easy to use, but create an interface between you and complex data. You learn a lot from them, and you become an expert of things that you would need to invest much more time to become experts of without. So we continue to push Anvi'o using our research. For instance, Florent Turgodet is studying phase variation, protein structure from pangenomics, and we're implementing tools in Anvi'o to do this research as he makes progress in this. Alex, a PhD student in our group, studies pangenome graphs for comparative genomics. Katie studies rapid evolution in the environment through DGRs and other means, and she's implementing a DGR profiler, for instance, into Anvi'o. Mete, who was a technician in our group, has been studying protein structure from pangenomics and large language models. Eva, you will hear from, studies metabolic dependencies, function annotation, how to improve those things in genomics. Sam, another person you will hear from, together with Eva, studies metabolic reaction networks in an attempt to get closer to biogeochemistry and start making ourselves relevant, and help our colleagues who study molecular formula and environments, not through sequences, but also from their chemical profiling. And Sam is focusing on transferring transcript sequencing and profiling. And all these things turn into tools in Anvi'o. And I will give you a very quick example using Alex's research, because I find this quite interesting and exciting. And these are so new as results that I thought, among all the things, I could perhaps provide you with some insights into what's coming in the context of pangenome graphs. So I don't know if you're familiar with pangenomics, but pangenomics is a means by which we can partition the gene pool of a bunch of genomes into separate bins, such as those genes that occur in every single genome and those that are occurring in a single genome, or those that occur in a few of them, like accessory gene clusters, core gene clusters, or singletons. And this shows a pangenome analysis of one of SAR11 clades, Amplus pelagibacter. And these are very close to genomes, and every layer in this display represents a genome. And every single item, and there are a few thousands of them here, these black marks indicate the presence of a gene cluster which means if it's a core gene cluster, every single genome contributed a gene because there were homologous genes that appeared to be in every genome. And if you are looking at something like this, like 100 of genes in this particular genome, whichever this one is, was only in this particular genome. I hope that makes some sense. It is a way to do comparative genomics at the level of gene classes by only focusing on sequence homology. You can see these things as de novo gene families. So here's a bunch of functions that are only encoded by this genome, and here a bunch of functions that are encoded by every single genome, and so on and so forth. So one problem with pangenomics is that it does not convey any information about the organization of genes in the genomic context whatsoever. So these core genes are next to one another in this display because they are clustered based on their distribution patterns, and we know that. So this region of this dendrogram, this hierarchical clustering dendrogram, represents those genes. But which ones of them were next to one another, for instance, in individual genomes? We have no idea about that. Is the synteny conserved? We don't know that. Or these single copy, singleton gene classes that appear to be nowhere else and only on this genome, or nowhere else only on this genome, are they occasionally next to one another, or are they uniformly distributed throughout the genome? These are interesting questions from the evolutionary point of view. Also, from the point of view of understanding how metabolic modules may be working. Do they have identical copies of every single gene, or some genes are variable across different members of close-rate populations? These are important questions. And without a solution that allows us to represent the connectivity in this display as a graph, it is impossible to approximate. So Alex has been working on it. And now we have a new Anvi'o tool. It takes a pangenome like this and creates this display. And here you are looking at a pangenome graph. It is no longer representing individual gene clusters as a function of their relevance to distribution patterns and how similar they are to other things, but they show as a graph how individual gene clusters form syntenies with one another. So it is kind of difficult to show this without the interactive interface, because in the interactive interface, we're able to zoom in, click on these individual nodes, see their functions and what metabolic modules they belong and so on. But here it's kind of complex. But what I want to show is a very simple example, for instance. We see interesting patterns like this one here. If I were to zoom into that, so this shows this part of the graph. Every single genome contains these gene clusters to be identical to one another, or extremely highly homologous. And there is one gene cluster here that seem to be different for almost every single genome. So if I were to focus on that region a little more specifically here, this is the overall genomic context. All genomes agree. Every single gene clusters you see here occur in every single genome. But this one here in particular has such distinct sequence similarity that average amino acid identity for these genes, even though they are at an extremely specific location, never exceed 20%. So what I'm telling you is that aliens come to Earth and realize that every single human being we capture from Earth have a hand and an arm, and then in between them, sometimes we see these watches that seem to be very different from one another. All hands are similar, and they are here. And then there are these watches that differ between individuals. And then there is the elbow, which is similar across everyone. This is an extremely important insight to understand certain phenomena about the relevance of watches and different kinds of those watches to be relevant to particular activities and so on. So this metabolic module happens to be a place where this organism is encoding its lipopolysaccharide, the cell wall associated function. I am not sure actually exactly what's happening here. But when we ask the question, so all these genes that are very specific in their location in the genome and very different in the sequence space, can we see whether their structural properties are conserved? One of the things now we can do is to take these sequences and ask AlphaFold to predict these structures, and then we can overlay these structures to see the extent of homology. As I mentioned, these sequences are quite distinct from one another, but in fact, when we look at their structures, this is what we see. This is many sequences, so one sequence from each gene cluster that differs in this place, overlaid, and you see a very actually conserved structural makeup. So this indicates that whatever these things bind, they are probably binding in a general sense the same kind of molecules, or they serve the same kind of function, but with slight differences. The prediction tools are not ready to be able to resolve those subtle differences, but this to me is very exciting. And then, for instance, this is another region, and this happens all over and over again. This, for instance, here, a bunch of genes that are very variable from one another in the sequence space, but their locations are very conserved, and their structures are very conserved as well, and so on and so forth. So we see this over and over again. And this is very soon going to be available to all Anvi'o users who are using Anvi'o to study pangenomes to be able to apply to their pangenomics artifacts out of the box without having to do anything else. So when I say we continue using our research to push the boundaries of Anvi'o is exactly this. PhD students in our group or postdocs try to advance their research, but they put the time into also adding those tools through which they advance their research into Anvi'o in an integrated fashion. And I'm there just to facilitate that and make sure that things don't go haywire. Which becomes harder and harder as I age. So I'm hoping that someone else will take the flag at some point, but so far, so good. And apart from reliance on our own research to push the boundaries of Anvi'o, we also rely on our community to continue developing Anvi'o towards the right direction. At the beginning, there weren't many people, but as of today, there are many people who contributed to Anvi'o code base and continue to do so. All these individuals, and there are more, are making sure that our code base is in good shape and bugs are fixed and so on and so forth. And they literally put their hands under the rug to help Anvi'o go forward. And in addition to that, with the recognition that Anvi'o has a very steep learning curve, we have been giving these free workshops all around the world. In fact, we trained around 700 scientists over the last 10 years, especially in pre-COVID times, but we recently restarted these efforts, and this Anvi'o workshop with a very large participation of individuals from all over the place took place in our new building at the HIFMB in Germany just a few weeks ago. And we continue to do these things, and anyone who is interested in on a very intense one-week workshop to learn more about Anvi'o and its application to omics data, they are welcome to get in touch with us and get a spot in these workshops. In addition to these workshops, we try to share our expertise and understanding of things with the Anvi'o community through this Discord. Currently has more than 950 members and over 3,000 discussion threads. And there's a link. If you go to anvio.org, you will find the link, and you click on that, and you end up in the Discord channel, and you ask your question, and you often get an answer if Florian and Eva are around and they're in the right mental space and have time. Or I try to do my best to contribute, even though, again, as an aging professor, my contributions are becoming more and more limited, unfortunately. But there are lots of individuals in this community that help each other, and that's very encouraging. In addition to these discussions and workshops, we literally rely on our community to implement new features in Anvi'o. For instance, people ask for things, and then we create these pull requests to propose new features to advance Anvi'o, from some very essential features to more recent ideas by suggestions from our colleagues. For instance, in this case, this is Julian and Jessica Mark Walsh telling us you can do better with calculation of coverage, and then we implement it in Anvi'o. And another example that will bring me closer to my final suggestion to this community, we recently got into ancient DNA stuff. I had no idea about ancient DNA, limitations, opportunities, and so on. But we started working with Eske Villarsev's group in Denmark through Antonio Fernandez Guerra, who works with him there. And he is using Anvi'o for ancient DNA data analysis. And he said, look, we are looking at single nucleotide variants in ancient DNA, but it is extremely difficult because of the fragmentation patterns, because of the deamination. Can we add something in Anvi'o profile so that it can help us rely only on variants if they are occurring in the middle of our short reads rather than its extremities, because extremities of ancient DNA is more prone to deamination. So create these variants that are not real. And then we worked with Antonio to implement an entire new feature. And then he tested it to tell us, okay, you did it right. I tested it and it's working. And that's the way we wish to work with new communities, because we don't want to implement tools just because they sound cool, which is the general computer scientist approach. You do things because they sound cool rather than you do things because they will be useful. But we wish to invest our efforts into things that are more likely going to be useful. So it is not a pipe dream of a computer scientist because it's technically challenging or it's cool, but because it's going to directly influence a life scientist's approach to their own data. And another example, again, with ancient DNA, it's extremely difficult to reconstruct genomes from ancient DNA. And we have this Anvi'o estimate metabolism framework that requires a contextDB, that requires a good assembly. And Antonio said, look, can I not give you my enzymes that I observe in my ancient DNA data, and I can run maybe Anvi'o estimate metabolism on them without having to give any genome, so I can still get metabolic completion estimates and so on and so forth. And then we implemented it working with them. So all these things to say that the Anvi'o community is a significant driver on new directions Anvi'o is able to shift its steering. Therefore, we need input. And when it comes to biogeochemistry, that input is, of course, generally lacking in our lives. But I'll give you an example of how we are slowly stepping into it. Jessica Fissel, who is also on the call, a postdoctoral researcher at the ICBM here in Oldenburg, has been studying biogeochemistry for a long time, and she was also interested in application of Anvi'o to her problems. And she generated this figure using Anvi'o, which shows the distribution of molecular formula in different combinations of cultures of microbial strains, which I'll try to give you a very brief idea about. So they conducted this experiment. There are 4 strains, relatively close related Roseobacter strains from marine environments, and they generated cultures with, for instance, 1 strain, or the mixture of 2 of them, or the mixture of another 2, or the mixture of 3, or by putting all of them together and so on. And they use the same exact artificial seawater medium with glucose as the sole carbon source. So the same medium, and then they observe the growth patterns, and then they sent these to FTI CRMS. I'm not sure if the machine looks like this. This is what ChatGPT thinks. I'm sorry if this is inaccurate. And then they generated these molecular formula. And I will show you one example to describe what that larger figure shows, because it's kind of a complex figure. For instance, this is an example of the combination of SH4 and SH24, these 2 strains. And this is the molecular formula they recovered when they grew SH4 alone. And this is the list of molecular formula they were able to recover when they grew SH24 alone. And this is what they got from the culture in which these 2 organisms grew together. And then the next 3 lines show what is unchanged from SH4 to SH4 plus SH24. What is novel that did not occur in SH4, but emerged when neither on SH4 or SH24, but emerged when they were grown together. And this shows what seems to be consumed by the combination of the 2 organisms. So then there are these additional layers. For instance, predicted molecular formula count for each of these culture petri dishes, and relative change between novel, consumed, and unchanged molecular formula, and then the number of strains, individualization, as well as the relative abundance of these individual populations in mixed cultures. So all that information is presented in this fashion, and then if you understood this, you understand the rest, and this has been done with many cultures in different combinations, obviously. And I'm not going to ask you to interpret this figure now, but I will share with you what Jessica shared with me regarding the overarching insights that emerge from this visualization in this study. And that includes the fact that consortium-specific molecular fingerprints emerge. So molecular fingerprints are not necessarily predictable, but they are specific to each consortia. Increasing number of strains lead to non-additive production of molecules. The example I gave was a great example for that. The number of strains increased from 1 to 2, but the number of metabolites are not additive. As you realize, predicted molecular formula count is not following the increasing number of strains. And in fact, there is an increasing number of strains leading to decreasing molecular formula counts due to consumption. Of course, when you create different combinations, they have different relationships regarding how they consume each other's products. And Jessica observes a nonlinear relationship between the relative abundance of strains and their impact on molecular composition, where this is a very interesting observation, because one strain, for instance, SH4, that typically occurs in this, it's always with this, it's shown with this color, it almost always dominates these co-cultures, but the relatively less abundant isolates influence the composition and molecular diversity that are seen by growing these things together quite substantially. So this, for instance, already uses Anvi'o for an application of these kinds of analyses. We understand none of it on the Anvi'o developer side, but now I know that there are new tools we can add to this framework that could make Jessica's work much more effective, and it perhaps would translate to your work if you're interested in similar things that Anvi'o could help, at least at the visualization stage. This, what I'm showing you, is an interactive visualization. These samples or a molecular formula could be clustered in different ways using different algorithms. You can make selections, you can put them in bins and annotate your visualizations, and so on and so forth. So I hope this part, at least to a degree, clarified the ways by which we rely on our community to continue developing Anvi'o. And I hope the talks by Eva Vesely and Samuel Miller in a few weeks will create a little more understanding about the potential things you could do with this platform and how you could benefit from it, at least currently, to study pangenomes, phylogenomics, or metagenomes, and so on, to integrate it into your workflows. So if you would like to see a little more about Anvi'o, what it is, and the list of programs, you can always visit anvio.org. And with that, I want to thank you all very much, and thank our support for Anvi'o development came from a few foundations. Lately, it's on the shoulders of the German taxpayers, and as well as CCOMP, an NSF initiative, a Science and Technology Center to study chemical currencies of our microbial planet. And Anvi'o is here thanks to all these people. If you were to visit this URL, you will see a list of developers and contributors to it. And I want to thank you very much for your time and your attention to this talk.
Awesome. Well, thanks so much, Meren, for the very nice overview and presentation of Anvi'o, and yeah, for building the tool and sharing it so openly with everybody. This is pretty awesome. Yeah, I think with that we're gonna move in a second to the Q&A, but I'm gonna stop the recording and just wanna say goodbye to everybody who's heading out. As Meren mentioned, in I think 6 weeks, we will have 2 more people from the Anvi'o team presenting a little bit more on the pathway mapping side. So if you're interested in that, feel free to stop by. And then before, we will have seminars on biosynthetic gene cluster maps, as well as multiple spectrum alignment. So yeah, hope to see you all next time, and thanks for stopping by. Okay.